start this seminar by giving a brief presentation of our guest today, and I'm gonna do it in Spanish, Professor. I'm gonna present you to the public, and para todos los que están conectados y escuchando. Buenas tardes a todos, bienvenidos a este seminario del Departamento de Farmacia. En esta ocasión nos acompaña vía Zoom desde Alemania el profesor Jonathan Gershenson, y me voy a permitir leer una breve semblanza del profesor. El profesor es biólogo graduado con honores en la Universidad de Carolina, Santa Cruz, en Estados Unidos. Realizó sus estudios doctorales en botánica en la Universidad de Texas, también en Estados Unidos, y comenzó su carrera en investigación y docencia en el Departamento de Botánica de esa universidad, para posteriormente trasladarse al Instituto de Química Biológica de la Universidad Estatal de Washington en Estados Unidos, y desde 1997 forma parte del Departamento de Bioquímica del Instituto Max Planck en Ecología en Alemania, del cual es director desde esa fecha. También desde 1999 es profesor honorario de la Universidad Friedrich Schiller en esa ciudad alemana, y es revisor científico en revistas especializadas de las áreas de botánica, bioquímica, ecología y química de alimentos, y también hace revisiones de fisiología de plantas, entre otras, y es miembro de varias asociaciones internacionales de biología de plantas y de fitoquímica. Y a lo largo de su fructífera carrera, el doctor Gershenson ha recibido múltiples reconocimientos, entre los que se destacan haber recibido una beca por la National Science Foundation y la Robert A. Welch Graduate Fellow en la Universidad de Texas, haber recibido la Harman Rimmer Lecturer por la Universidad Estatal de Washington, y estar dentro del top 10 de los autores más citados de ciencias de plantas en Europa. Y además recientemente ha sido reconocido como uno de los mejores científicos de plantas en Alemania, donde ocupa la posición 4, y en el mundo, donde ocupa el sitio 22, de acuerdo con el número de publicaciones y citas. Y a propósito, el doctor Gershenson ha publicado cerca de 500 artículos, los cuales abordan la bioquímica de los metabolitos secundarios de plantas y sus mecanismos de acción en herbívoros, así como la evolución y regulación del metabolismo secundario. Y la mayoría de su trabajo se enfoca en dos grupos de metabolitos secundarios, los terpenoides y los glucosinolatos, de los cuales precisamente nos hablará el día de hoy en su seminario. So after this presentation, Professor, I thank you again for being here with us, and we are really anxious to hear you talk about this interesting topic called plant toxin and insect detoxification, the chemical basis of a natural equilibrium. So the audience is here for you, Professor. Thanks a lot for accepting this invitation.
Thank you very much for the invitation, Bernice, and I also should thank Jennifer Munoz Gomez for also being part of the invitation committee. It's really a special pleasure to talk to you and try and tell you a little bit about some of the work that we're doing here in Germany. If there's a technical problem or something, please let me know. And or if I start talking too fast in English, just scream and I'll try and slow down I sometimes get a little bit carried away. But it's it's already evening for me here, so hopefully things will go at a nice slow pace. Let me start out by saying that I work at an institute that's really devoted to chemical ecology. And when you think about chemical ecology, you can imagine that it's actually a rather broad topic. Almost any interaction between two organisms can somehow be a chemical interaction. But our focus here in our institute is really about interactions between insects and plants. And in my group, we're especially interested in how plants protect themselves against insects and how insects get around these protections and still feed on plants. When you walk outside, you see plants. Well, maybe not now in Germany as many as you would see in Mexico, but there are lots of green plants out there, and you still see insects. So clearly, neither side is really winning completely. We still have insects and plants. And what we're curious about really is some of the biochemical and chemical mechanisms that are responsible for what I call this equilibrium. And what I want to tell you today about then is about a system that we've been working on for quite a while, a system that involves one type of plant toxin called glucosinolates. And this is a group of chemical compounds that are found in the Brassicaceae, the cabbage family, and a number of related plant families. I'm sure you've eaten them and tasted them before. Let me show you a little bit about the chemistry. I know you want as many chemical structures as possible, so I've tried to pack a few of them in. So here's a basic structure of a glucosinolate. You can see that the center is really this carbon-nitrogen double bond, this imine function, and then there's a thioglucose residue and a second sulfur as part of a sulfate group here attached to the nitrogen. So it's an N-oxime sulfate, really, with a glucosal residue. And the R group then varies depending on the amino acid that's responsible for the biosynthesis. I can tell you that here's a structure of a complete glucosinolate. This is one called glucoraphanin. It's one of the major anti-cancer glucosinolates. It's been studied quite a bit. It gives you an example of what a full compound looks like. And you can see the sulfur atoms over here, and we even have a third sulfur here in this side chain. So one of the main things to tell you about glucosinolates so that you're really try and understand them is that these are what we call activated defenses or two-component defenses. If you look at the structure of the parent glucosinolate over here, these are actually not biologically active themselves. They require first a second component, and the second component is an enzyme that cleaves off the sugar. And so this is a glucohydrolase enzyme that cleaves the sugar, and we end up with an aglycone without the sugar. And this enzyme is a glucohydrolase, is usually called myrosinase for glucosinolates. And it's essential for cleaving this hydrolyzing this sulfur glucose bond and producing an end product here. And this is actually not an end product. This aglycone can rearrange to form a variety of different compounds. The most common toxic derivative are compounds called isothiocyanates. And if you're chemically interested here, you can probably see that this is a rearrangement. The R group has moved from this carbon here to the nitrogen. So we have RNCS right here. This is an isothiocyanate. And these are one of the main groups of hydrolysis products of the glucosinolates, and they're quite toxic to most organisms. Now, in the plant, these isothiocyanates don't form spontaneously. In the plant, the two parts of the components of the system, the glucosinolate and the hydrolyzing enzyme, the myrosinase, are actually present in two different compartments. And so you don't see this reaction forming. But when the plant is damaged by like an insect, for instance, then the two compartments combine and we get this hydrolysis. So this is the story of the activated defenses, the two-component plant defenses, that you don't see them until the plant is actually damaged, and then a toxic product is formed. So this is true for other compounds as well. Things you may have heard of cyanogenic glycosides, which release cyanide. It's a rather similar system like the glucosinolates. Again, a glycoside is present that gets hydrolyzed to an aglycone and releases the active compound. But now I want to tell you a little bit more about glucosinolates. And in terms of human society, I told you they're present in cabbages. You've all tasted them before, maybe even for lunch today, or will. They're present in radishes, broccoli, cauliflower, cabbages, kale, Brussels sprouts, all of the vegetable crops of the Brassicaceae that you're probably used to having. And they're the main flavor principles of all these compounds. The glucosinolates give rise to these hydrolysis products that give us the distinct flavor of all these different vegetables. They're also present in a number of condiments. For instance, mustard, the sharp taste of mustard is an isothiocyanate that comes from the glucosinolates. Horseradish, which is popular in Germany, and I guess rábano picante is popular in parts of Mexico too as well. And so this also has glucosinolates in it, and also wasabi, which you know from Japanese cooking, Japanese cuisine. The active principle there is also an isothiocyanate that's derived from a glucosinolate. So these are part of our lives in more ways than one. Here is a field of rapeseed or canola, and here these plants contain glucosinolates. When the farmers harvest the oil, they want to make a little more money. They like to take the seed cake that's left behind and sell that for animal feed. But if it's high in glucosinolates, it's poisonous to many animals. So one of the things that in breeding rapeseed that farmers and agricultural people have done is to try and reduce the level of glucosinolates in the seeds, but keep them in the leaves because they help protect the plants from insects. So this is another example of glucosinolates being involved in human society. Here they're bad. The farmers don't want to see them in their rapeseed, in the rapeseed themselves. They just want to squeeze them and get the oil out. And then finally, another area that's very interesting in terms of glucosinolates has been research on cancer prevention. You may have heard over the years that it's very good to eat broccoli, very healthy because of the glucosinolates. These are considered to be very effective against a variety of different kinds of stomach cancer, especially intestinal cancer. And people are still studying how the value, and there's a generally message that we should be increasing the level of these in our diets, and that will prevent a number of different kinds of cancers. So that's a little bit of an introduction to glucosinolates in human society. Let me tell you more about the insect and plant world then. So what I want to do is tell you two kinds of stories today about some of the research we're doing. The first is about the toxicity of the glucosinolates to the insects. And so this are talking about these plant compounds and how and why they're toxic to glucosinolates, to insects, herbivores. And then secondly, tell you a little bit about how insect herbivores can get around this, how a number of insects have developed methods to feed on plants containing these toxins without getting poisoned. So I'll tell you these two different stories, and then start out with the toxicity story. And this is, I chose this one for you because as pharmacists, you do care about. Sometimes biologists say, okay, it's toxic, and that's the end of the story. But for me, and hopefully for you, you care about the mode of action. Why are these compounds toxic? What is the mechanism for toxicity? And I'll tell you a little bit about that today, tell you a little bit about what's known. So here is what you've already seen. Here's the glucosinolate. It gets hydrolyzed, the sugar gets cleaved to get this aglycone, and that rearranges to form an isothiocyanate. And if you look at this isothiocyanate from a chemist's perspective, you might notice that this carbon atom here, which is double bonded to sulfur and to nitrogen, to two heteroatoms, is very electrophilic. And this is the source, really, of the reactivity of this molecule. And so if you look in the textbooks, they will tell you that isothiocyanates are toxic because they combine with a variety of biological nucleophiles. So we have a very electrophilic carbon. It can react with biological nucleophiles, such as the cysteine residues of proteins or lysine residue, where you have a nucleophilic residue that's exposed, and form these conjugates. And these are, this is considered to be the basis for toxicity. So by the reaction of isothiocyanates with proteins, we then inactivate sulfhydryl groups, which are often responsible for catalysis. But when we went ahead and did this experiment here, in which we radiolabeled glucosinolates and we fed them to insects, and we looked to see where the radiolabel ended up. only maximum of about 20% ended up in the protein fraction, these black. So these are different insects that we fed this radioactive glucosinolates to. And there really wasn't much label in the proteins. So this suggested that the isothiocyanates might be doing something else than just binding to proteins, as the textbooks told us. So what we then did is look to see what they were reacting with. And it turns out that the major compound reacting with isothiocyanates is a compound called glutathione. And you might have heard of this. This compound is present in all eukaryotes and most prokaryotes as well. This is a tripeptide of glucine, cysteine, and glycine, and it is a major compound involved in redox control in all cells. It reacts very readily. Here's a very nucleophilic sulfhydryl group then here, and it helps control, really helps control the redox state of cells, and it's present in millimolar amounts sometimes, so fairly high amounts. So we found that the isothiocyanates had often reacted with glutathione to form these glutathione isothiocyanate conjugates. Again, a very electrophilic carbon here reacting with a nucleophilic sulfur to form this conjugate right here. And so how could glucosinolates be toxic then by reacting with glutathione? Well, what we did is we fed insects with isothiocyanates, and we found, as you might guess from something like this, that their level of glutathione was could decrease very strongly when feeding on glucosinolate-controlled diets. So here on the right, we have insects that fed on this. This is an isothiocyanate, ITC. Here, that's actually the one I just showed you already from the glucosinolate I showed you. And you can see that when insects feed on this versus a control diet, this is a couple of percent of isothiocyanate, which is fairly normal for a plant diet. You can see that they could lose over 90% of the glutathione content is gone. And that also has consequences for the rest of metabolism. If we look here, these are the 3 amino acids of glutathione, that tripeptide that reacts with them. You can see that the glycine and glutamate are not significantly changed, but the cysteine is way down. So you have less than half of the amount of cysteine present in an insect that is feeding on a diet high in glucosinolates or in these hydrolysis products, these isothiocyanates. So that might not be good for the insect. And in fact, I'll show you here some data. This is growth curves of insects that have fed on glucosinolates. And the upper 2 traces show insects that have fed on either 0 glucosinolates, a control, or 1 millimolar isothiocyanates. And they're generally gaining weight here. This is a little bit irregular because they molt every now and then, which is a little annoying, and they lose a little weight. But if you see below here, this is 4 millimolars isothiocyanate, which is the leaves of Arabidopsis, say, or cabbages, or somewhere between, produce 2 and 4 millimolar isothiocyanate. And if you feed insects on levels of glucosinolates like that, you can see that the growth rate can be much, much lower. And as it turned out, you can also see a decline in protein, which is probably the most revealing statistic. So one of the reasons they're probably not gaining weight is they just don't have as much protein on these higher glucosinolate diets. And what we think is happening is they run out of cysteine because they keep having to make more and more glutathione to conjugate with the glucosinolates. And they run out of cysteine, and therefore they just can't make as much protein. And that's what might cause this problem. So this is sort of our explanation now for why feeding insects on glucosinolates might cause reductions in growth, why this serves as a toxin, because it's actually depleting the amount of protein, of cysteine available, and which eventually causes them to have much, much lower growth rates. But there may be other explanations that we've looked into, and let me show you a little bit about one of these others that's been done by a student that's currently in my laboratory. And what he did was he decided to look for other possible targets for isothiocyanates in insects. And as we do this these days, what he did was he used a non-targeted metahomelomics method. So he took an insect to which he fed glucosinolates to, and he took an insect which had no glucosinolates, and then he went ahead and he compared the metabolomes. And this is a so-called volcano plot, which you might be familiar with, shows the difference in metabolite. We're plotting at the bottom here on the x-axis the degree of change in different compounds. So these are all increasing toward this side, decreasing toward this side. The y-axis shows the significance. And this compound over here got our interest because it's decreased here by a factor. So this is log base 2 here. So this is a factor of -10 log base 2. So this is hundreds of fold decrease in this particular molecule. And the student here, Tom Duke Mai here, went ahead and analyzed this compound to figure out what it was from mass spectrometry. And he figured out that the structure was an enzyme cofactor called tetrahydrobiopterin here. This is a cofactor of certain enzymes involved in amino acid hydroxylation. And he actually found that the isothiocyanate right here could be conjugated directly to this compound. We found the conjugates and inactivating it. So this might be responsible for why there's so little in it. And then he wondered whether this could also cause toxicity. So we have a compound here that we had no idea that Tom found for us that is actually reacting with these isothiocyanates. And he wondered what could happen. So we knew that this was a possible cofactor for aromatic amino acid hydroxylases, such as the enzymes that convert phenylalanine to tyrosine here on this side here. And I'll switch the picture here to the other side so you can see here. So we have phenylalanine converted to tyrosine by tetrahydrobiopterin, and then that can be go on and hydroxylated again to form dopamine. And what Tom showed is that insects feeding on glucosinolate-containing diets then show a drop, not in phenylalanine, but significant drop in tyrosine. And likely because of this cofactor is then disappearing by reacting with the glucosinolate hydrolysis product. And we also have a lot less dopamine. So feeding on glucosinolate-containing diets may also cause trouble in these compounds. Also serotonin, which you might know is a good neurotransmitter, a derivative of tryptophan, is also in this category. And what Tom did is he went on to show why this could be important in insects. You might know that insects, very important to insects is making a cuticle. The cuticle is very high in derivatives of tyrosine, amino acid, from the amino acid from tyrosine. So he actually showed that insects feeding on diet had lower amounts of, basically had a thinner cuticle. I showed you that had lower amounts of tyrosine and dopamine. And this picture is trying to show you that they actually had the cuticle looks redder, which means that it's actually thinner. So an insect that has fed on a glucosinolate-containing diet then has a thinner cuticle because it just doesn't have as much tyrosine. Another toxic consequence of feeding on glucosinolate-containing diets is also an increase in DNA damage. And here, one of the metabolites of the tetrabiopterin, the cofactor that Tom identified, is deoxyguanosine, which is a precursor then of DNA. And what Tom was able to show here, and what you're doing is looking at DNA that's been analyzed on an agarose gel and then stained. And this tail, so the DNA is moving electrophoretically, and you can see this big tail behind it here, shows the extent to which the DNA has been broken. The tail of this comet in this assay actually gets bigger and bigger and bigger. So from below, you can see measurements of the tail length. This is a measure of breakage in DNA strands. And you can see this increases here with more glucosinolates that have been with more glucosinolates that have been in the diet. And so this suggests that this is also a sign of toxicity of the glucosinolates. Again, DNA breakage, thinner cuticle. So these are physiological problems that insects can have on feeding too much on glucosinolate-containing diets. So as a sort of a conclusion to this part then, what I could tell you is the textbooks about binding of these isothiocyanates to proteins don't seem to be correct. What we found is really glucosinolates bind to this glutathione, which then causes this depletion in cysteine. And then there's also evidence that there may be other targets of isothiocyanates in insects. And we still have a lot to learn, I think, to figure out what the targets. For most plant compounds that people study, most of these toxins, we have no idea. We barely know a lot about mode of action of human toxins in many cases, but in insects, we know even less. So very often we know that compounds are toxic, but we really just don't know why. And this is some research which we really wanted to show exactly why this compound might be toxic and why it might cause insects to die when feeding on such plants. So I've told you a little bit about, okay, here are the pictures and the levels of glucosinolates. So I've told you then about the toxicity of glucosinolates. And now in the second part of the talk, then I want to tell you about how insects can get around this. So we all know that when we walk outside, we see insects feeding on plants. And many times they feed on plants that we think of as toxic. So how can they do this? I've just told you how toxic glucosinolates can be. If you've tried to grow cabbages, you probably know that their insects will come and feed on them. I'm trying to think of what the big pests would be in Mexico, but there could be many pests if you try and feed on cabbages, try and grow cabbages in your garden. And you might wonder, if this plant is so well defended, why are insects able to feed? So we also wondered about this when we started this work a few years ago. We were interested in knowing how insects kind of evade these sorts of toxins. So let me start out by just telling you a little bit about herbivores in general. When we think of insects or mammals that feed on plants, we usually divide them into two classes, generalists and specialists. The generalists, like this grasshopper or a moose, they're considered to be herbivores that feed on a wide variety of plants, and they avoid toxins. They're thought to just feed a little bit here, a little bit there. If something is too toxic, they move on. But there's a second group of herbivores called specialists, and you might know a lot about this one here, the koala bear, which eats only eucalyptus. Pandas eat only bamboo. There are many insects that actually also specialize on just a few plant species. And these insects are considered, and these, like koala bears too, are considered to have very specialized guts that are adapted to feed on eucalyptus or adapted to feed on some toxic plant. And so that's why that's what they do. They're very well adapted, but maybe not to other things. So we wanted to find out what the secrets were then of insects feeding on glucosinolate-containing plants. And if you look at glucosinolate-containing plants like the Brassicaceae, here are some examples of some of these insects. They actually are a number of specialists. You might have seen this one if you've ever tried to grow cabbages. This is the cabbage butterfly. It's a white butterfly. They have caterpillars that are can kill all your cabbages very quickly. In Germany here and in Europe, there are a number of different herbivores that are very specialist. We have an aphid, we have a little flea beetle here, we have this hymenopteran here, we have other caterpillars that feed only on glucosinolate-containing plants. So how do they do this? So here I've come back to this same diagram again. We have the glucosinolate, which you might remember, hydrolyzed to an aglycone here, and then forming and rearranging to form an isothiocyanate. So what can an insect do to block this activation of defense? So one of the tricks that we found is that they convert the glucosinolate, they can metabolize it by taking off the sulfate and making what we call a desulfoglucosinolate. So we have, instead of an NO sulfate, we have an NOH here, this aldoxine moiety. And it turns out this could be a good trick because this compound is not recognized by this enzyme. So it's not converted to an aglycone, and therefore you don't get this rearrangement into an isothiocyanate. So if an insect could convert glucosinolates into desulfoglucosinolates, it might be able to avoid formation of isothiocyanates. And there have been some reports in the literature about this. And the first report was really for a caterpillar, Plutella xylostella. And over the years, we've added some other, there's some locusts which do this and feed on glucosinolate-containing plants, and a few other insects that actually can also create desulfoglucosinolates from glucosinolates. So this were some nice reports in the literature, but what we realized a few years ago is that we didn't quite know why these are effective. So an insect that could feed on glucosinolate-containing plants, is this sulfatase really working faster than the hydrolysis of glucosinolates to block this hydrolysis? Maybe there was another mechanism that we had been missing. So to show that this was really a detoxification, we worked a little bit on Plutella xylostella. This is an insect called the diamondback moth. You might have heard of it also from an agricultural point of view. It's specialist pest on the Brassicaceae. It's actually considered to be the worst in the world. It's undoubtedly all over in Mexico too. It's especially in tropical areas or subtropical areas, very, very common on lots of different brassica crops. In India, it's currently a huge problem on the brassica species as well. So what we did to try and understand whether this reaction is really a detoxification is we decided to try and knock it out genetically. Okay, I'm sorry. Let me first show you that some of the biochemical data. When we looked at what compounds were formed when we fed labeled glucosinolates, here is the diamondback moth. I had shown you these upper 4 bars before. Diamondback moth is very unique in that almost everything it produces are these desulfoglucosinolates shown in green here. So they actually have a very unique metabolism of glucosinolates. You can see they make a lot of it. Of the label we added, about 80% that was turned into desulfoglucosinolates. And this suggested that this detoxification mechanism was probably working because the biochemistry, we just didn't see glucosinolates. We saw most of the label was present as these desulfo compounds. So we went ahead to try and confirm this by knocking out the detoxification system. We had the genes involved for this. We had shown the enzyme activity. Genes have been identified from Plutella. And what we did is we took this detoxification gene, the gene that encodes this sulfatase, and we transformed it into a Arabidopsis here. And we transformed a piece of the gene as a double-stranded RNA construct. And the idea was we wanted the insect to feed on the Arabidopsis, ingest the RNA, and then this would silence the gene. So instead of silencing it directly in the insect, we're using the plant to produce a silencing construct, which then goes into the insect and silences this detoxification enzyme. And we tried to do this in 2 different plant backgrounds. One was an Arabidopsis that contained glucosinolates, and the other did not have aliphatic glucosinolates, which is the major group in Arabidopsis. And so we made these transgenic lines and fed them to the diamondback moth. And what you can see here from the data is, first of all, here we have the transcript levels, and you can see that the silencing worked very well. In the background with glucosinolates, we can reduce the transcript by over 95% here. So we've silenced the gene that's supposedly involved in this detoxification. We got actually less silencing when there were no glucosinolates. That's because the expression is much lower of the gene to begin with. And then if we look at the enzyme activity, you can see that in the silence lines, we got less than half of the sulfatase enzyme activity that we did in the wild type control. So the silencing worked in that we were able to reduce the transcript and reduce the enzyme activity. So what happened in the insect then? We've knocked down this activity. What is happening metabolically? So here is what I've shown you before. Here's a glucosinolate being hydrolyzed to an isothiocyanate. And you remember that in this Plutella xylostella, they are making this desulfo compound. And if the insect is doing well and things are working, most of the glucosinolate should go this way to the desulfo compound and very little to the isothiocyanate. But we come in there and we silence this gene now. So what should happen? In theory, we should push flux over to this side and get more isothiocyanates. And that's exactly what happened. When we did this silencing, we actually decreased the desulfo profile by 90%, and we increased isothiocyanates by tenfold here. So the silencing actually worked at the level not only of the gene, at the biochemistry. We were blocking this detoxification pathway. So now we can see, is it really a detoxification or not? But the real test then is to look at the insect and say, is it doing better or worse? So let me start here on the right side. And this is a measurement. These are the best data, so I've got to show them to you first. This is pupal mortality of this insect. How well does the pupil survive? And we're comparing here the wild type here. These are plants with glucosinolates. And RNAi is interference RNA. If we silence this detoxification enzyme, we increase pupal mortality by 4-fold. So this was pretty striking. We're able to kill 4 times as many insects if they don't have this detoxification enzyme present here. And this is the control without the enzyme. Over on the right, this is the controls without any glucosinolates in the background. And you can see they survive fairly well. And then over here on the left side, we're looking at larval weight, which is another way of showing how well the insects are doing, their performance. And you can see that the larvae doing the worst, again, this gray one, are the ones that have the enzyme knocked down, interfered with, and feeding on a diet with glucosinolates. This line here is the unsilenced line, and then the top 2 are ones without any glucosinolate at all, and they do the best. So what this data really showed us nicely is that this particular enzyme reaction is really, we could call it a detoxification activity because it really silences it changes the fitness, the performance really of the enzymes on this particular diet. And actually, interestingly, I should come back to this data here. If they have no glucosinolates, they do even better. But interestingly, in nature, this insect will never feed on a plant without its glucosinolates. It will never lay its eggs on it, and it refuses to feed on it because it's a specialist, and it knows that it should be on a glucosinolate-containing plant. But it's interesting to see that if it doesn't have any glucosinolates, it actually does even better. So we can discuss a little bit more about that later, about how specialist insects work. But what I hope to show you here is that this is a real detoxification activity. It can make a difference in insect performance. And I have another example here for you, which is similar in that here again, you see the glucosinolate being hydrolyzed to the isothiocyanate. And here, instead of desulfating, there are some insects which add an extra sugar here to the glucosinolate. Here we have 1 sugar and adding a second sugar. And it turns out that this also prevents this compound from being a substrate for the myrosinase, and you end up with, you block this particular conversion here. You don't get any isothiocyanate because this compound is glucosylated, it just doesn't ever get hydrolyzed. And it turns out that we found this extra glucosinolates with the extra glucosylated glucosinolates in a number of different insects. And these are all phloem feeders. If you know insects, aphids, whiteflies, and mealybugs all feed on the phloem of plants. And we never found this in leaf chewers, only in phloem feeders. And what we were interested in again, was finding the gene to study out the evolution and whether this was fitness. But we looked at this and we realized that we had an enzyme that was putting on an extra sugar. We needed a gene that encoded such an enzyme. So to find this, what we did is we actually did some labeling experiments with labeled sugars to see if they would end up on glucosinolates. And if you know glucosinolate metabolism or even human metabolism, you might expect that an enzyme that adds glucose like this is glucosyltransferase. There are a number of enzymes that add sugars to toxins, which make them less toxic. More polar, sometimes that helps in excretion. But when we added labeled glucose, we never found glucose added to the glucosinolate that was labeled. So we went back and we actually used disaccharides. We actually used labeled sucrose here. This red indicates the label. These are carbon-13 labels that we've used to label the sucrose. And here's the glucosinolate. And this actually worked. When we labeled the sucrose, here's the glucose moiety, here's the fructose moiety. You can see that the extra sugar of the glucosinolate, here's the glucosinolate right here, here's the first sugar. The second sugar is labeled from sucrose. So sucrose worked, glucose didn't. And we can also label sucrose just on the glucose part, and that worked. We also had our label. If we labeled sucrose just on the fructose part, we didn't get any label in our product. So it became clear to us that the enzyme that was putting this sugar on is an enzyme using sucrose and then using the glucose part of sucrose to label the compound. And it turns out there are such enzymes present in insects. These are enzymes called transglucohydrolases. So they're from the GH13 family, if you're familiar with the CAZ nomenclatures. They're glucohydrolases that actually cleave sucrose, and the glucose moiety becomes enzyme bound and then is transferred to other residues like this one. And interestingly, this was a transglucosidase. And so we were able to then use this to look for the enzyme activity. And finally, we were able to find the gene involved in this process in whiteflies. And the reaction is the glucosinolate is then converted to, from addition of sucrose, to have a glucosylated glucosinolate by a transglucosidase. And this is then inactive. So whiteflies are very big pests on some Brassicaceae plants. And this seems to be the trick of how they do it. They add an extra sugar, and that gives a product which is non-toxic. And we were curious about the evolution of these enzymes because when we looked at the sequence here, it turned out that these are very close to other enzymes in aphids that are involved in converting two sucrose residues to higher order saccharides. For instance, a reaction here in which this sucrose is split, and this sucrose gets added to another sucrose, and we end up with a trisaccharide. So aphids actually do this quite a lot. Aphids, you might know, if you know about aphids, they feed on phloem, and often they dealing with sucrose concentrations of 1 molar or higher. So these can be very desiccating. There's a lot of osmotic strength, and this is thought to be one of the ways in which aphids cope with it by adding, by making higher order saccharides, trisaccharides from sucrose. And so if you keep adding, you can make multiple residues here, and this ends up having product which has less osmotic activity for the aphid. So it turns out that the enzymes that were involved in this reaction here seem to have been recruited to be involved in this detoxification enzyme. So it's a nice little story which told us how they might have evolved this particular activity. So I think I am close to finishing here. What I want to do is, okay, I'll go. Here's just, we've isolated many other different metabolites from glucosinolates. I've told you about desulfo compounds. I've told you about glycosylated glucosinolates. I've showed you a little bit about, so these both block the formation of isothiocyanates. I told you that isothiocyanate can react with glutathione. This is actually a detoxification mechanism too. You can imagine some insects might do this, but some insects, and this is what we do as humans actually, how we detoxify isothiocyanates. We let this toxic product form, and then we convert it into a glutathione derivative. And there's a whole pathway involved in recycling some of the amino acids, and we end up with an acetylcysteine as part of the pathway. So there are a number of derivatives which we've been able to isolate that are involved in glucosinolate detoxification mechanisms. As a general principle, some of them convert. The specialist insects seem to have the best system where they metabolize the glucosinolate before it is actually hydrolyzed to form isothiocyanates. One of the specialists actually diverts the hydrolysis to the nitrile, which is not toxic. Other insects, and other herbivores like humans, we let the isothiocyanate form and then conjugate the formed isothiocyanate. The danger here, of course, is that we are releasing a toxic product, and it might not just bind to glutathione, it might bind to a sensitive target site. So a variety of different ways in which insects have really developed to metabolize glucosinolates. And sometimes when you look at all these methods, you wonder why these compounds are actually toxic. Another thing we can do is also inhibit the glucosidase itself, to the enzyme that activates. That's also another mechanism. So what I've tried to do then is show you a little bit of a balance, why some plant compounds can be toxic and how some insects get around this. I think hopefully you've gotten these major points. These glucosinolates are specialized toxins in the cabbage family. The toxicity that I've showed you appears to happen because we lose the glutathione. The reaction, glucosinolates react with glutathione, the hydrolysis products, and we end up with lower amount of cysteine. But what we have shown in the last research in the last year or so is that there are other targets in the plant. I told you a little bit about tetrahydrobiopterin, which is a cofactor for some of the aromatic amino acid hydroxylases that might be responsible. So reaction of glucosinolates with this cofactor might also be responsible for some of the toxic effects. And then, so that's on the plant side, and on the insect side, I told you a little bit about how ways in which insects can get around these toxins. And basically, it's by metabolism, their own metabolism. They can do things like desulfate or add extra sugars. And by knocking one of these out, we showed that it really was a detoxification process. So we have a situation in which we have plants making toxins and we have insects that get around these toxins. What does that mean in the long run? And that's kind of why I called it an equilibrium, I think. In nature, we're just starting to study this, and these interactions have been going on for, in the case of glucosinolates, close to 100 million years since glucosinolates were invented by plants. So there have been 100 million years in which insects could be selected for various adaptations to get around these toxins. So in some sense, we shouldn't be surprised that there's so many insects that actually can feed on these plants. They've adapted to it. Why don't all insects feed on these plants? Well, presumably there are some costs to these. And we saw that a little bit in the fact that the insects that didn't produce any, once you silence them and they didn't have any glucosinolates, they were actually better off. So presumably there are advantages to detoxifying. You can feed on plants, but if you don't have to do it, you can also save energy as well. And maybe this kind of arrangement is what's caused the situation we see today, in which we have toxins out there, but we also have many insects that are developed the ability to feed on toxic plants without suffering much damage. So many have specialized detoxification systems. So probably at the end, I should also tell you, though, that detoxification is not the only way for insects to get around toxins. And you've probably heard of transporters before. This is another form, and human adaptations to toxins. In many cases, we have transporters involved too. That's a whole other story of how by transporting toxins out of the body, we can also avoid them. And another adaptation really is by changing the actual target, so where it's not susceptible to toxins. So there are a number of different approaches that herbivores have used over the years to adapt against various toxins. Humans we are also a herbivorous creature. We spend a lot of time on eating plants and plant material. We also have a lot of different adaptations to feeding on toxic plants. So that means not all toxic plants are safe. Some of them we're really adapted to fairly well. Some of them Obviously not. So after millions of years, we still have plants and we still have organisms that feed on them. And there's sort of a general chemical equilibrium of these. And I hope you can now appreciate that and maybe appreciate some of the biochemical mechanisms behind it. So thanks for your attention. Let me just acknowledge some of the people who did the work. Here are pictures of the 4 main contributors to the stories I've told you. 3 wonderful graduate students, Verena, Michael Ruo, and Daniel Vassau is a postdoc who really supervised a lot of the work that I've shown you today. Postdoc from Brazil, who's still here in Germany, and they were responsible for a lot of the work. Some other people were involved as well, and I also need to thank the funding agencies for their support, and also thank you for your attention. So thank you. Okay. Sure. I'm happy to stop sharing and then you can, yeah, welcome to ask whatever questions you want.
They are kind of a shy audience, Professor. But
Okay.
I have a question for this kind of enzymes. I don't know if you have considered if climate change might have an impact on this kind of detoxification processes, because the climate is getting warmer, and I don't know if the desulfatation processes are like a specific thing, or maybe this can impact in other species of insects.
Yeah, no, that's a good topic to bring up. Thank you. That's on everyone's mind too. I think that, you know, a few degrees of temperature probably won't make a difference in the activity of the enzymes, but the range of the insects may change quite a bit. So insects like Plutella, which are subtropical, are supposedly moving north because of climate change. So, so that's certainly changing the balance between them as an agricultural pest. You can imagine also that climate change could affect the levels of glucosinolates or maybe the levels of other compounds in the plant, which might make a difference for the detox, the outcome of the plant and the insect together. So I think you're absolutely right. There could be some changes involved in that, and I think we're still wondering. You know, many people, not only climate change but higher CO2. Will probably have a change on plant chemistry. Plants sometimes grow better under higher CO2. The only experiments with glucosinolates suggest that higher CO2, they have less glucosinolates. So that might end up making a little bit of a problem too for when that's involved in plant defense. So I think there are a lot of things to consider when we talk about effects of climate change. And I think there's a lot to figure out from both the plant side and the insect side.
So you can see her.
You don't need to put that. No, I can see. Thank you very much for your impressive talk. I really enjoyed that. I was wondering if what you mentioned about glutathione, you see that when the toxin is forms the thiocyanates, and the thiocyanates affect the glutathione. Do you see that? Properties that had been attributed for many, many years to this family, the Brassicaceae, and know. So I was wondering about that. Do you know any?
Yeah, no, that's also a very good question. I think there have been different opinions, and currently what people think is that it's really the isothiocyanates are actually acting by a signaling process. So it's not directly by glutathione derivative, but actually the isothiocyanate changes. So there are a number of signaling proteins. There are a number of proteins in the so-called keep domain, which have a family which are involved in signal transduction for various detoxification processes. And what's believed to happen, so the glutathione might be involved because there is a change in redox state that may trigger the activity of this signaling pathway. But what happens is you eventually the body actually defends itself better against a variety of different reactive oxygen species, and that's considered to be one of the main ways in which glucosinolates then enhance defenses against cancer. So considered to be through the oxidative stress pathway and by actually altering the levels of a variety of different defense enzymes transcriptionally. So rather than direct through glutathione, that's the current opinion. There've been a lot of work, and there may be more than one mode of action for this, but there may be, this is a reactive compound, as I tried to show you. There is very reactive electrophile. We don't yet know all the targets of it, but that's the current consensus.
She used to work to protect crops using canola, canola oil, and so, well, very nice. This is another, another view of evolution, right? It's very interesting. Well, thank you very much, indeed.
Thanks a lot, Professor, for your answers and your talk. I think we don't have many more questions. So we thank you again for your presence in here. Y les agradecemos a todos que hayan venido al seminario y nos vemos regresando de vacaciones. No, esa no es. No, una indicación de que alguien tenía algo, pero no. I'm checking. Yes, I'm checking here in the Zoom chat. They have a question. Arumir Lira, if you have a question.
Can you hear me?
Yes, we hear you.
Okay. Yeah, I have a question regarding agriculture, as in cauliflower or broccoli that you mentioned in the beginning. Do you think knowing the mechanisms of detoxification of these insects, it's possible to develop an insecticide that can silence or block any of these insect shields against the natural toxicity of the plant?
Yeah, I think that what people are, there's currently a lot of interest in India because of if this is a big pest on rapeseed in India, and the Plutella xylostella that I talked about with you in India, and to a lesser extent in China. So what they are trying are ideas about the interference RNA. So they're using this to see if you could spray this in the field. This is supposed to be environmentally friendly. It's just, DNA. It breaks down very quickly. And that can, if it can be taken up, in theory, it should be efficient at silencing insects. So that's being tested. There's also a group testing a specific inhibitor of the sulfatase, testing that to see whether that could be applied as a pesticide. So, in modern agriculture, we don't want to just spray and kill all the crops because, as you know, you do more damage by killing the enemies of the insect. And so they really want a method that will target the diamondback moth. And that's why the idea to use the if this gene that's involved in the sulfatase, this is a very specific enzyme only found in this insect. And so if we can target that very specifically, that might be a good control. So yeah, so thanks for that question. I think it does fit within modern approaches of being very specific and targeting individual pests. And it may be a situation like in India where they still have such a high value crop, in this case, Brassicaceae, which is the Indian rapeseed, which is really susceptible to damage from insects like this, that they may be able to really be motivated to try and find targets. And so this might work. It hasn't, nothing really being used commercially in the rest of the world that's kind of based on this kind of work. Farmers will just use standard insecticides right now on Plutella. But it's developed resistance to many of the insecticides. It's also developed resistance to Bt toxin. So it's one of the more dangerous insects out there. It seems to be able to get around a lot of things so that people are interested in trying new methods to find resistance. I hope I answered your question.
Yeah, thank you.
Bueno, entonces, ahora sí damos por finalizada esta plática. Muchas gracias por su presencia, profesor. Once again, thanks a lot for your talk.
Gracias.
Está bien raro cómo lo conectan.